welcome back to my channel Back and By Wall. In today's video, I'll be discussing high fidelity DNA polymerases. Firstly, let's get into the basics. Suppose this is a genome of an organism which I'm studying. Okay, and I want to amplify this red region, that is these particular genes, that is B, C, D, E, F. I want to amplify these genes, okay, so as to go further for my analysis, for my study. What I do, I synthesize a set of primers, usually a forward and a reverse primer for this specific region, okay, flanking this specific region, and I add a polymerase, okay, I add a polymerase. Okay, it can be any polymerase, TAC or some high fidelity DNA polymerase, which I'll be talking further. I add a polymerase which amplify, or which we can say extends the 3' dash OH or forward or reverse primer, and as a result of which I get a number of copies of that particular region. So I can further study for my purpose, for my goal, whatever I want to study. Okay, this all is done in a thermocycler, which is known as polymerase chain reaction. Okay, polymerase chain reaction here means polymerase means I use an enzyme polymerase. A chain reaction means that is the product of first reaction is a reactant of the second reaction. Okay, so as to get a number of copies I want to study, I go for polymerase chain reaction. I synthesize the primers which are flanking that particular region of interest, and as a result I get number of copies and I can go further for the analysis. Sometimes what happens, I want to study a specific, we can say, polymorphism, or I want to go for the cloning. Suppose I want to we can say insert an X gene into a particular organism. I need that X gene to be very accurate. Like if the sequence is ATGC, I have to make sure that the sequence is the same when I insert it into a vector for the cloning. Otherwise, if it is not the same, what happens when the replication takes place, there will be an alteration of the codon, there will be alteration in the formation of the protein. So I need to have a specific sequence, which I can say the specific desired sequence which I want to insert in vector should be very accurate. Or in some other things like which I used to do in molecular biology, I used to study the single nucleotide polymorphism. By single nucleotide polymorphism means that there is an alteration at one point. Like in this example, which I'm highlighting, a C is transition to a T. Okay, a C is transition to a T. So as to get this result, okay, so as to get these accurate result, I'll be requiring a DNA polymerase which is having high fidelity. Okay, these enzymes, the high fidelity DNA polymerases, are the enzymes that offer high levels of accuracy. Okay, for molecular applications, which I've already given two examples, SNP studies and cloning, which require a low error DNA replication. Okay, why high fidelity DNA polymerases? It is what it is modified version of a conventional PCR in which a DNA polymerase of high fidelity, or I can say a low error rate, is used so as to get the exact, or we can say the accurate results of what is happening in the, or I can say what is there in the genome. Okay, and examples I've already told you: SNP studies and cloning, expression analysis, sequencing, genotyping, and some of the examples which I've mentioned in this slide of high fidelity DNA polymerase. First is the fusion high fidelity DNA polymerase, and in the bracket I have mentioned the times which is higher in compared to that of fidelity of TAC. TAC polymerase is a general, we can say a thermostable polymerase which we usually use in the PCR reactions in our day-to-day laboratory techniques. And but in case if I require results very accurately, I switch to high fidelity DNA polymerases, and the examples are fusion, which is 50 times higher fidelity than TAC; Q5 high fidelity DNA polymerase, approximately 280 times higher than TAC; platinum super phi 2 DNA polymerase high fidelity PCR enzyme with approximately greater than 300 times stack fidelity, acuprime TMP-X DNA polymerase 26 times than TAC, PFU DNA polymerase, and code FX neum. Okay, these are some of the examples of the high fidelity DNA polymerases. That was all about today's video. Thank you so much for watching my channel. Please like, subscribe and share if you like my content. Thank you.